So now we've looked at the mappings in IGV. Let's look at the program that we can use to refine that data and automatically look for variations for us. The program we're focusing on in this course is called Freebase. There are other snip calling tools. For example, the most well-known might be GATK. But let's use Freebase here because it's a one-liner and it's a bit simpler pipeline. So Freebase is just a normal program, and as an input, remember input is orange, output is green here. Input, we need our reference sequence, and we can add several BAM files. So this could be grain.bam and leaf.bam and so on. And then we redirect that into a so-called VCF file, variant call format. So again, as a little exercise, we have our BAM files now. Let's have a look if we can run Freebase on all our three BAM files, and then check how that file is looking. Go back to my virtual machine to get rid of IGV. Okay, so ls BAM, these are my BAM files, and I want to run Freebase. I need a reference, and then I just can use leaf.bam, grain.bam, root.bam. And if I just run that, it would be displayed on the terminal, but I redirect that immediately into a file. Let's call that tissues.vcf. Might take a little bit of time to run. And let us inspect this VCF file. So at the beginning, we have lots of lines starting with 2 hashes. So these are our header lines that explain most of our fields. And then we have one line starting with 1 hash. And then here we also have headers. So this is quite important, and we'll see later why. This tells us the headers of the columns coming below. However, something's still a little bit wrong here. This one is called unknown, so something doesn't work. And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different columns here. So here, this is straightforward. Again, we have our chromosome, we have a position, and then we have some identifier we can enter. This seems to all be a dot, so this field is not used by Freebase. We have a reference allele, an alternative allele, some quality. Then we have a column where filters that might be added would be entered. So here we have dots. At this point, we haven't filtered our VCF file at all. And then we have an info column, which is similar to what we've seen before with GFF formats. So this is a column where you can just dump a lot of information, and these are these kind of key value things. So we have an AB field, and then equals and some value. And then this is semicolon separated. And if we move further, we will come to a column called format, and then we come to a column called unknown. So let's see where our format column is. We move through, there's a lots of data dumped, and here's one column. So this is a format column. And now what happens here is we have some additional data column. So the idea here is that in this format column, you have different fields that are colon separated. And here in this next column, or what we see in a bit, that all the columns that come after that, we have the same number of fields as in this format. So the GT field here is the first field here. The second field here is DP for depth. And here we can see the same. The 4 refers to depth. So all these columns that come after the format field, at the moment we see only one. All these columns have the same field headers. Okay. So if we go back to our presentation, we pretty much went through that. And again, here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 columns. So here, this seems already to work a little bit better, because here we have, the idea here is that we have one column per sample that we entered for our SNP calling. We talked about all these things, including the info field. And here we have a format field which says genotype, GQ, DP, HQ. So 4 fields. And for each of the samples coming, we have the 1, 2, 3, 4 different entries. Another interesting thing is here in that header row, instead of 10, what we saw before, here we have 11. So that's for every sample. And in this header, you will always have the sample ID. Now, when we just used our freebase call, there was only one column following the format column, and it was meant unknown. The problem is that there wasn't enough information in our mapping data, in our BAM files, that satisfies this need. So basically, this means we need to redo our mapping. And while we are redoing our mapping, we need to enter some additional information, which means we need to add a read group ID. So this read group information, this is the normal HiSat2 call, which we did just a minute ago. HiSat2, then we give the index 1, 2, our reads, and this is our output file. And then we would add this additional 2 parameters, so minus RGID, and then we give it some ID, and then minus read group, and then in double quotes we have SM, for that is our sample name, and then this input thing, this orange part here, is what's going in the header column in our VCF file. So let's try to regenerate that for our leaf and grain data. Okay, so if we go back here, we need to rerun our HiSat2 call. So at this point, for example, if we use grain, we need to add minus RGID in quotes grain. I just call the ID and the sample the same. So here we need to call it sample, so SM colon grain. So we run this, and we would need to do the same. I just do it for 2 samples now. Okay, unfortunately, we need to do our SEM tools again on that. SEM tools sort minus O grain.bam grain.sem. SEM tools index grain.bam, and the same for leaves. Okay, so at this point we can then run. Let's just do it for the last sample as well. We run HiSet2 on root. So then we have all the data for the 3 samples together. And we can use the same freebase call again. Okay, let's see if I find my freebase again. So here we have our BAM files that I just recreated, including the read group now. We need to rerun that again. And hopefully our tissues.vcf file now includes the proper headers and also more than one column after the format field. Okay, we go down to our column, and you can already see here in this line after the format field we have leaf, grain, root. And now if we move into the first line, here we would see GT, DP, AD, and so these are all the fields. This is our format column, and then this is followed by 3 additional columns, which are the 3 samples that we entered. So if you consider this VCF file, this GT field is one that is most commonly present whenever you do a SNP calling. And this is our allele call. So here we have 2 values here, 0, 0. So this is assuming that our sample is diploid. So it can have one of the 2 values. And 0/0 would mean this is homozygous because it's the same. And also 0 because it's the very same as the reference. Same here. So the next tissue is also 0, 0. And what is our last? This is also 0, 0. So the genotype call that has been made, although we see some variation here, is always this is most likely the same as the reference. We can look further down. For example, here at this position, there's a different call. There's 0, 1. So this means that the genotype call is guessing that we have heterozygosity here at that sample, which might be right or wrong. Most likely this is a wrong call or a false positive call, because also if you look at the second field here, that's DP, the depth is 3. So we have only 3 reads supporting this call, and therefore we probably are not interested in that. However, if you pass these things, you might want to filter a little bit for depth, but the genotype call here is what you're most often interested in when you look at these VCF files. Okay. So let's go back to our presentation. Just to recapitulate this field, the VCF format, we have meta information lines. They start with 2 hashes. This provides all the information about the program that was used to create it, the format that we're using, and all the input data. Then we have this 1 header line, which contains our columns. We have 10 plus column, so everything after the 10th column contains our sample names. And then we have all the data lines. We've went through that. If you want to know more about the VCF field, this is the link, which you can just use to read the entire specification of VCF. Okay, so we learned how to add the read group information. And there's something similar in BWA if you're interested in that. You just would need to look at BWA, and there's also an option how to add read group information. Okay, so now thinking back at the overall project that we had been looking at, we were interested in the Kariha genome, and we were set out to find a resistance gene in there. So in the RNA-seq course, we looked at RNA-seq data, saw if any in any of the tissues something is further or more expressed than the others. That didn't get us very far. So now we can look at SNP data. So we have just mapped Kariha data against Kariha, which doesn't help us a lot. But how about we use some RNA-seq data from an accession that does not carry this resistance gene? So in fact, avoset S is susceptible. So let's take this data and we use HISA2 mapping. We want to add a nice read group, and we run freebase to call these SNPs that we have. We want to inspect that data. We can use that, we can load the BAM files into IGV. Interestingly enough, you can also load VCF files into IGV. So let's give that a try and see what we find. And the idea is to now finally look at SNPs, and you need to find out which is the resistance gene. So to get the final outcome of the whole exercise, what are the positions of the resistance gene in the Kariha genome? For this one, you might want to think back to BLAST, how that works, and find the gene. So let's do this exercise together. What is the first thing we need to do? We run freebase using the avoset S data, and just for practicing, we go and try to add this read group here. So we need to add the flags -RGID, myID, or whatever ID we want to add, and then we actually add this read group sample. So let's run this here. And I think we have the data of avoset here, data, raw data. I've copied it here. If you haven't it here, you need to import that onto your VM now. I go into my exercise folder. Maybe I can clear up everything that has been here before. It's nice and empty. I do need my reference, of course, in this folder. So I copy data reference locus.faster into this, and this is RNA-seq data. So of course we're using HISA2, and we need to create this HISA2 index again, although we probably have done that just now. HISA2 minus build index. You probably have all this right now. So we've created the index files, and now we need to copy our avoset raw data into this folder. I have it in my raw data folder. Okay, so now running HISA2, we need to remember the read group sample to add. So HISA2 X, we've called the index here just index for IDX. If you have many indices in your working directory, you probably want to give that a more unique name. Something a bit bigger. So what else do we need to add? We need to add the --RGID. And we need to add the --RG, which is SM colon, use here SM colon, and then give it avo S. Okay, let's see how that works. Now we need to generate the BAM file, SEM tools. This is just repeating the same thing over and over again to get used to it. -O. Because this is SEM tools sort. And we need to create an index on our BAM file. Okay, you probably have that already. If you don't have the faster index FAI file, you need to create that. So this is the FAI file, which is very often implicitly built by things like IGV, but it's good to have that explicitly done, then we know what's going on. Okay, maybe just very quickly, at this point we can inspect the data. Well, the general idea was to use FreeBase to call our snips. Maybe we do that first. FreeBase. Let's go back. What are the calls for that? We need to say -f and then our locus.faster. We give it a BAM file. We have only one at this stage. And then we redirect that into some VCF file. Okay, so we have a VCF file with our avoset snips. We can scroll down a bit further. Here, this is our read group sample that we have added. So you might remember I set the read group ID to avoset and the sample to avoset -s. So this is what's appearing here in our column header. And then we have a couple of snips. There's lots of information. This is probably all useless. We need to go to the end and look at our column. And here the first field in this is our genotype. So here we have a heterozygous snip. That's probably not what we're looking for. We're looking for fixed mutations. So you can already see in this file, it is really hard to figure that out. Maybe we can load that into a big text file or we can load it in an editor and view the text file in there, or we can load it into IGV. So let's just do that. Let's have a look at this data in IGV. Let's start IGV. Since we've used that before, our locus.faster immediately comes up. If that isn't here, we need to load it via load genome from file. And then select the locus. So I generated this faster index earlier. If that faster index file would not be existing right now, IGV would generate that implicitly. So there we can just load it, and then we can load different tracks. For once, I'm interested in the gene annotation, just to look at that. I've downloaded that and put that into references Cariha version 1.locus.gff3. So I've got the gene annotation. I load the actual mapping itself just for our convenience, that we can inspect what's going on. And I can load the VCF file. Okay, so here from the VCF file, you see where we have SNPs called. We have the general track and then the AVOZS specific track, which is, since this is the only sample, it's a little bit redundant information. So we have a few SNPs here, and then we could zoom in, could have a look how many of those are actually in a gene or in an exon. So here you can see we have two SNPs that are within the exon of a gene. You can see here that ends with LC, so this is a low confidence gene. You can also see that these SNPs here, if we hover over them, this hovering is this, I've set that to hover. If we don't want to have this tooltip popping up, then you can either never show or show and click. Right now we're interested in that information, so let's hover over them. And you can see here we have a depth of 2. And the same is true here. This pretty much matches what we can see here in the reads. So if we would load the alignment track on top of that, we can see we have only 2 reads here. So that's not a lot of statistical information to call a SNP here. So that might just as well be some sort of wrong mapping. Okay, so what else is there? Let's zoom out a little bit further. What is obviously an immediately interesting locus is this one in the middle. It seems to be a very long gene. So we have a lot of SNPs here, and they are all in an exon. By the way, you can see these things here. If we right click and expand this, you can see that we have one low confidence gene. Let's get rid of the tooltip for a bit. We have one low confidence gene here. And these intron exon structure seems to match very nicely how the RNA-seq data is mapping. We have some additional low confidence genes annotated here, but they are not of any interest for us at the moment. So if we go a little bit further in here, look at these SNPs. I'll get my tooltip back. You can see here we have a lot of depth, 520 reads matching at this position, and they really support a different allele. You can see this is a homozygous perfect SNP. And if we go through this exon, we can see that there's a lot of difference between those things. Okay, so we could obviously inspect every other gene in this region, but this is a very strong candidate here. What we can do from here, of course, we could just take this gene. This seems to be most of the coding region. And then with the region navigator, can add this as a region, right click and copy the sequence. And if we're interested, we could now use our Firefox or genome internet browser that hopefully starts at one point. And blast this sequence against non-redundant proteins that are on NCBI. Let's search for a blast X. Can paste the sequence I've copied out of IGV. And we're blasting here against non-redundant protein sequences and see what comes up. Okay, so we got a result. We have cut most of the waiting time out of our video. But so yeah, we get some results back. The first is whole genome shotgun wheat. I'm not going into that. The next one is NLR protein, so nucleotide binding site in leucine-rich repeat protein, LR13. And hopefully if you have looked at the Carija paper, you can see that the YR27 that was cloned in that paper was a homolog of this LR13. So having this as one of the best hits is a very nice candidate to be our disease resistance gene that we were looking for. So I hope in this course you have learned a little bit how to deal with sequence data. You have learned how to blast, you have learned how to map data and do sequence alignments, a little bit of SNP calling. Most importantly, everything you do, try to load into IGV and visualize it, especially when you go into your final loci that you're inspecting. You will see errors in your analysis and you will be able to just find candidates much quicker if you look at your data rather than just running pipelines. So at this point, it might make sense to go back into that YR27 paper and have a look what of these tools have been used. Yeah, and with that, we finished the sequence analysis course.